So how in the world is it possible that you can see things? Well, the story begins with you eating some carrots. Carrots and other vegetables contain lots of molecules called carotenoids, such as beta carotene. Here's a structure for beta carotene. As you can see, it's kind of long. I didn't feel like drawing it myself. In your body, these carotenoids get broken down into vitamin A, also called retinol. As you can see, it's almost like we took beta carotene and cut it in half. When I say the word retinol, you might be thinking about skin creams. And the interesting thing about bioactive molecules, like vitamins, is that they have many different functions in your body. That is, you need them to see, and they can also help your skin look better. But for your eyes, this retinol needs to be turned into retinal. And as you can see, all of the double bonds are in this trans position. So we're going to call this all trans retinal. Enzymes in your retinal pigment epithelium, or RPE, are going to bend this. So they're going to take it from all trans to 11 cis retinal. And as you can see on this 11th carbon, you're changing the shape of the retinal. And a photon of light can come in and change it back. Once you have your 11 cis retinal, you can covalently bind it to an opsin protein. As you can see down here, this is happening at the amino group. This is called a shift-based linkage, if you need some good trivia. And together, the 11 cis retinal and the opsin form your photoreceptive pigment. This, in the end, is how you see. There's 4 different types of pigments. One called rhodopsin, found in your rods, responsible for your nighttime vision and black and white. There's 3 different opsins found in cone cells, receptive to 3 different wavelengths of light. And one cone cell will only express one type of opsin protein. A lot of this knowledge of retinol and retinal and these pigments is brought to you by George Wald, and many other scientists, of course. But he's very notable for his advances in visual physiology, right? So he studied pigments in the retina, discovered vitamin A there, and what it's doing. You look up on Google and Wikipedia, these are some notable things about him. And I drew very heavily from his paper he wrote in 1968 after winning the Nobel Prize on his work. So thank you to scientists like George Wald and many others who studied vision, because without them we wouldn't know jack. Let's move on and talk about rhodopsin, the complex now formed when the chromophore is bound to the opsin protein. When light comes in and hits our 11 cis retinal, remember this is our chromophore, it's going to bend about that 11 cis position back into the all trans form. This process is called photoisomerization, or basically just changing the shape because of light. When this happens, our chromophore is going to get kicked out of the opsin protein back into the cytoplasm, and it's going to cause a change in shape in our rhodopsin. Another way to visualize this is in the plasma membrane itself. You can see here we've got our plasma membrane in yellow, our rhodopsin in purple, and our chromophore visible in black. Remember that when light hits it, it's going to strain it up and kick it out. And so what that's going to do is activate a new protein, 3 subunits, called transducin. One interesting fun fact I want to point out before we continue is that this whole process, phototransduction, is actually one of the most well-characterized G protein coupled receptors in biology, or GPCRs. My classmates who have recently taken cell biology may remember GPCRs and how convoluted their function is. Anyhow, transducin is our G protein, that is to say it's got GDP on it normally when it's inactivated. Here's the structure of GDP down here, but when it's activated by rhodopsin, it's going to drop the GDP in exchange for a GTP. And in this process, it is actually going to dislodge this alpha subunit here on the right from the other 2 subunits. And then the alpha subunit with its GTP is going to go activate something else. We can see that down here with all of our alpha subunits and our cGMP phosphodiesterase. The alpha subunits are going to activate our cGMP phosphodiesterase. cGMP stands for cyclic GMP, guanosine monophosphate. And so as you can see, the phosphate is bound to 2 of the oxygens on the guanosine here. One of those bonds is going to get cleaved by this enzyme and turn it into GMP. One really key point I want to emphasize before I move on is the purpose behind these G protein coupled receptors. Why bother going through so many steps to activate one enzyme? This isn't the only place they're found, by the way. They're all over your nervous system. And the reason for this is because they amplify the original signal, the original stimulus. So in this system, you can see that we have 1,11 cis retinal getting turned into all trans retinal, of course. You're activating one rhodopsin. This one rhodopsin can activate many, many, many transducins. Those many activated alpha subunits of the transducins can go and then activate a bunch of this enzyme, cyclic GMP phosphodiesterase. At the end of the day, you want this because that is how you get big macro scale change in the cell. Well, all right. I mean, why do we care that cyclic GMP is getting decyclized? Well, that's because in the dark, in your rods, there's a bunch of cyclic GMP gated ion channels that are forced open by cyclic GMP. When they're open, you have a bunch of sodium flowing into the rod cell, causing a dark current. It's kind of a silly name, but what they're really describing is just the constant influx of sodium ions into the rods. When you decrease the levels of GMP or cyclic GMP, after light hits rhodopsin, now you're closing this gated ion channel and there's no longer going to be sodium flowing into the cell. Okay, so why do we care about sodium flow anyhow? Well, let's take a look at a neuron here in the dark. A rod cell, going to be tightly brushed up against a retinal pigment epithelial cell, RPE for short. It's going to come in handy later. We're going to talk about the RPE cells. But if you remember, when you're in the dark, these rod cells are actually depolarized, meaning that sodium is flowing in because the gated channels with CGMP, they have CGMP. So sodium is flowing into the cell, and this is increasing the charge of the cell, right? So the cell potential, the charge, the difference in charge outside and inside the cell is actually raising. So normally neurons like to sit at around -70 millivolts. When you increase the cell potential, that allows them to fire an action potential. They're going to send a signal. And so at the synaptic terminal, what signal is the rod sending? It is releasing lots and lots of glutamate. So glutamate is a neurotransmitter, and it's actually inhibitory. So the rod in the dark is depolarized. It's essentially activated, and it's actually inhibiting the cell that's attached to the synapse. I find this pretty interesting. It's really weird to think that like the photoreceptor is actually on technically all the time in the dark. And when it's hit by light, that's actually when it turns off, right? Because if you remember, in the light, now we have our photon. So our photon of light is going to come in actually from this direction. It's going to bounce around a little bit and then finally hit our rhodopsin. When it hits the rhodopsin, remember it triggers a bunch of steps, and that eventually leads to the CGMP gated ion channels to close. And so then sodium can't actually get in anymore. Sodium is not flowing in. When sodium is not flowing in, the potential is actually decreasing. So now you're decreasing the cell potential. Okay, that's great. When you decrease the cell potential, you can't fire your action potential any longer. So you're releasing less glutamate. It's actually a gradient, so they can activate in a gradient. So now you're releasing a lot less glutamate in the light, right? Depending on how much light is coming in. Now, when the cell that's attached to the synapse is getting way less glutamate, it's less inhibited, and therefore it can fire some signals, right? And so a little zoom in electron micrograph here of a rod here on the left. A cone is on the right. You can see these discs that I tried to illustrate here. They're really, really tightly compacted. You can imagine how much rhodopsin you can fit on there. It's kind of incredible. All right, so let's talk about the layers of cells in your retina, right? So we talked about specifically what's happening at the photoreceptor when light hits it, but then how does that go downstream turn into a signal to your brain? So let's go from deepest layer to most superficial. That is furthest back in the eye to furthest in the front, closest to the light. We're going to start back here with our retinal pigment epithelial cells and our rods. One thing I wanted to point out about the retinal pigment epithelium is that if you remember, the rods are actually kicking out the all-trans retinal. I had to make sure I was right there. They're kicking out the all-trans retinal after rhodopsin gets activated. And they're actually sending that to the retinal pigment epithelium. So I'm going to go all-trans retinal. This is going, well, to be more specific, retinol. Retinal is toxic, and so it gets rapidly reduced into retinol. That all-trans retinol gets sent to the retinal pigment epithelium. The retinal pigment epithelium, if you remember at the very beginning, it turns all-trans retinol back into 11-cis retinal. And so this regeneration process actually takes a bit of time. And so if you've ever noticed walking into a dark room after being inside all day, it takes a while for your eyes to adapt. It can take up to 40 minutes for this process to fully complete. And that's because in the daytime, your rods are actually inundated with so much light, they kick off all of their 11-cis retinal. They're completely bleached, is the word they use. And so you need time to regenerate all of your 11-cis retinal to see in the dark. On the other end of this rod cell is a bipolar cell. And so it's directly attached to the photoreceptor and then to a ganglia cell. And the ganglia cell is where you make a bunch of connections between different rods and cones. And then these ganglia cells connect to another layer of ganglia cells, another layer of ganglia cells, and eventually all of their axons line up into nerve tracts, and that gets sent to the optic nerve all the way to your brain. So the signal gets sent from your rod inhibiting the bipolar cell. When it cannot inhibit the bipolar cell any longer, the bipolar cell sends a signal. That signal goes to the ganglia cells, and that signal gets sent to all these folks. And then in combination, because remember, other rods may be inhibiting their bipolar cells, and so these ganglia cells are getting all kinds of information, will eventually get sent to your optic nerve to be interpreted by your brain as vision. All right, so here's a cross-section of an eyeball that I drew. I wanted to really emphasize that the photoreceptors are one of the deepest layers in your retina. I find this kind of counterintuitive and weird because it's like, dude, the light's coming in from the front. Wouldn't I want my photoreceptors to be closest to the light? But actually, that's not how it is in reality. Whether or not that's the most effective way, I'm not really sure. But that's the way that it is. And so some diseases, such as macular degeneration, you'll actually have damage to these deep layers of retinal pigment epithelium and the photoreceptors, of course. And when those layers are damaged, you can actually have issues of structural integrity of the retina itself, because the layers will start to fall in on themselves. Because as you can see, the retinal pigment epithelium and the rods actually hold up the rest of the layers. Here's a really cool scanning electron micrograph where you can see the cone cells in purple and rods in white. And you can tell this is probably a section of the peripheral retina because there's just so many more rods than there are cones.